This is TWIS. This Week in Science, episode number 1022, recorded on Wednesday, July 9th, 2025. What will we never know? Hey everyone, I'm Dr. Kiki, and tonight on the show, we will fill your head with exploding cucumbers, zoonoses, and sensitive crabs. But first, thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience. Disclaimer, disclaimer, disclaimer! Like the pains of an aged tennis player, so go these days of our lives. It hurts to get up in the morning. There might be grunting and groaning, but you know that to keep moving is to stay alive. So get up, get moving, and get ready for This Week in Science. Coming up next.
I've got the kind of mind that can't get enough. I wanna learn everything. I wanna fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I wanna know what's happening, what's happening, what's happening this week in science. What's happening, what's happening, what's happening this week in science.
Good science to you, Kiki.
And a good science to you too, Blair. And everyone out there, welcome to another episode of This Week in Science. We're back to talk about science. And yet again, there was a wonderfully wide world of wondrous, I can't use wonder over again. Anyway, so much for being not repetitive. But there was a lot of science this week, and so we dug through the stories and brought them to you. I have stories about some going back in time for zoonotic infections. What? That's so exciting. Um, Justin's not here tonight, so I might be able to chit chat a little bit about the complete human genome of an ancient Egyptian. And then I've got new brain cells, which is so good. And maybe some synchronized dance brains as well. What did you bring, Blair? What's in the animal corner?
Well, I have an appetizer of some plant news in the form of explosive seed dispersal. Then in the animal corner, I have sensitive crabs and emotional tortoises. I'm having a lot of feelings today. And then I saw that, Kiki, you brought an animal story for tonight. I did not get a chance to read it at all, so I cannot wait for you to tell me all about it.
I did read it a little, and I went, Blair, how could you not? I can't believe that this is a story, but it is. So chimpanzees, they're decorating themselves. Interestingly, we'll talk about that one. Okay. Everyone out there, if you would like to know more about past episodes, the present episode, once all of the stories get put up on our website, we will have show notes available, links to stories, other, our YouTube page. Thankfully, Fada put some great notes up about the sources of our stories. Same thing goes for our website, twist.org. Wherever you are, make sure you are subscribed to TWIS. If you're not, look for This Week in Science. Make sure that you're subscribed and you're sharing us with your friends so that everybody can have great science conversations and continue to learn new things, ask new questions, and develop their practiced and considered curiosity about the world. There's a difference between knowledge and wisdom. Yes, there is. Let's see. Anything else? No. So just subscribe. Look for This Week in Science. We're on YouTube, Facebook, Twitch. Twist.org is our website. It's time for the show. You ready for some science?
Bring me some science. I
will
demand it.
Okay. By demand of Blair, I bring you my opening story of the night, which actually is science I can't tell you about. because it's science that we may never actually, answer. There's questions that may never be answered, at least in the United States, by the researchers, that were involved in this particular event. So this week, an event was produced at Capitol Hill, and it was produced by the, Congressional Science, uh, panel. So, what is this? This is the event was from the House Committee on Science, Space, and Technology, and the Democrats in the Rayburn House office on yesterday, Tuesday, July 8th, from 11 a.m. to 2 p.m. on Capitol Hill. They hosted The Things We'll Never Know, a
Mm.
science fair of canceled grants. And so there were 20 or 30 or so researchers who represented their work and, the general science that has been impacted by budget cuts, by the cutting of grants due to words like diversity, equity, inclusion, people wondering, like one researcher wondering why her work studying cancer had been canceled. She's like, I don't really understand this. Another researcher who was reported by NPR, Sumit Chanda, professor of immunology and microbiology at Scripps, focuses on pandemics, preparing for disaster, and because of the budget cuts to science funding, Shonda, along with the other scientists that were there, presented the work that is potentially never going to be funded or answered, the questions that were involved related to pandemics and, preparation to those kinds of things. Um, these researchers were there to bring science and the important questions that have been stalled to the Capitol Hill, to be in front of not just other scientists, but in front of policymakers, in front of stakeholders, in front of the congressional representatives who are going to be voting tomorrow, Thursday, July 10th, on the budget for United States science for the next year. So it's very good timing. There's a lot of great science. Some Ars Technica reported on this, and unfortunately, there were actually several individuals who did not want to be reported on. Their names are not
Mhm.
into the public. And they're not even necessarily afraid of repercussions at the federal level. They're afraid of repercussions in their home states, which are
Hmm.
uh, have certain things going.
So
Yeah.
let me ask this. If I'm a researcher and I lost my grant, am I unemployed now?
No, not necessarily. It depends on your role at the university. Universities do
Right.
hire
Because you could be a professor
as a
also, or yeah.
with an with an annual salary, and you have various roles within that salaried position. But
Mm-hmm.
to, do the work you need to do, you need to be able to get a grant to pay your lab assistants. You need to be able to get funding to
Mm-hmm.
pay for the equipment in your lab. You need to be able to, um, have the funding that will allow you to even buy time on telescopes or buy,
Mm-hmm.
to share in some of the larger, more expensive technologies that are being used. And
Mm-hmm.
currently, this last week, a very interesting story popped out related to Harvard and some of the other, and this is not the only university, as technology transfer has been going on for a long time with intellectual property coming out of universities and going into startups and larger companies. It's a big money business. Uh, Harvard has had and some of the larger um, Ivy Leagues are looking at venture capital, private equity. Researchers are going out on their own to get private equity money to support their research.
Mm-hmm.
And then Retraction Watch. this week was like, oh yeah, so this one Harvard researcher who's getting I think I forget where it's coming from, but, he's getting private equity for the work that he's doing, and suddenly there are a whole bunch of questions about, reproducibility of his results, of
Mm.
falsification of images, of, you know, the
Keep your
molecules.
capitalism out of my science, please.
Right, like, at the beginning of like, there's stages,
Ugh.
right? It's how do you do the moonshot stuff without like, does everything have to have a purpose? And then also we know, like, if it isn't going well, when do they, what happens then, right? What happens to the
Mhm.
entire line of research if maybe it's not even falsification or other issues, but maybe they have the right question or the right tests have not been done right, but it's no longer worth the time, the money that's invested. Like, how do you keep that going? So,
Mhm.
relying solely on private money or majority on private money to allow for the advancement of science is putting, I don't know, backward steps.
Mhm.
It's taking big moves backwards.
This is not only is research not going to happen, but established laboratories will need to shut, which means they will have to be reopened when funding starts
Oh,
again, which means, yes, we are moving backwards. Like, there's going to be a huge amount of time wasted reestablishing
yep.
all of these labs and protocols and materials and animals. Like, any labs that have animals that they're going to have to
Yeah.
rehome or other things because there isn't funding to take care
Mm-mm.
of those individuals anymore. So then they're going to
And
have to start
that's a big
over.
deal. Yeah. Yeah. And it's,
Ugh.
of course, not all of the research. There's another paper out this week that is doing, they're analyzing the ecosystem of philanthropy and where money is coming from and how it's funding science. But the bottom line there is that, again, philanthropy is not enough to be able to take up, what the
Mhm.
government was doing. So anyway, Um, just to put it out there, there are some incredible researchers who are studying really important questions, and their work has been put by the wayside. And this week, our Congress people are supposed to go back to the voting about how much money to give to American science. So, hey, might be the last. If you haven't made a phone call yet, might consider it. Um,
Mm-mm-mm.
uh,
But
uh,
yeah, yeah, yep. Tax
um,
cuts for the extremely
yeah.
wealthy. Sure. Sure, sure, sure.
Because, we all wanna be there someday, and of course, right now, that's, yes, don't tax the rich because I'm gonna be rich someday, come
Right.
on.
Right.
I'll pay for other people to get rich
Right.
'cause no, wait, what? No. No.
Look around. You don't even own a house. Not you, this hypothetical person.
Ah. Okay, so that was my, explosion of where we are on the federal science front again for this week, and, I'm glad that there's a bit of it that's performative, but it's also educational, it's informational, it's getting the science in front of people, um, and hopefully getting them to understand why it's important. Why do we wanna study this gene? Oh my gosh, because after 200 publications, it might have cured childhood inherited deafness. Yeah, no.
Who needs it?
You know what I need? I
What?
I need you to tell me about, explosive seed dispersal, please.
I loved this story. Okay, so there are these things called squirting cucumbers, Ecbalium elaterium. They are loosely related to cucumbers. They're in the same family as like squash and pumpkins and stuff like that. But they perform explosive seed dispersal. So when you think about seed dispersal, you think of a few things. You think about like those seeds that have little helicopter wings that float to the ground. You think of some sort of wind-based seed dispersal like dandelions, right? You think about animal-based seed dispersal, which is, you know, when animals eat something and then they poop out the seeds, right? So there's all these different ways that plants have to get their seeds out there. They want it to go away from them, leave the nest, propagate the outside area.
And
So
not
there are
just depend on
some
the wind. Yeah.
there are some that are much more active. And so these cucumbers, these squirting cucumbers, quite literally explode. It's quite an explosion. I was hoping that you'd pull it up. They're also called touch-me-nots. And so Kiki right now is showing us slowed down footage of the seed dispersal. And you see a few seeds and then more, and then more, and then more. You're like, you got more seeds in there? Oh my gosh. There's so many seeds.
That's
just keep
amazing.
coming and coming and coming. So researchers used microcomputed tomography. They created a 3D model of the fruit, and then they also did micro CT imaging and high-speed videography to capture the exploding fruit in detail. So you just saw the high-speed videography. They recorded the explosion of the fruit with high-speed videos at 1,000 frames per second and 10,000 frames per second to calculate the speed of the seeds and the possible shooting distances. It happens so fast, naked eye, you're like, what happened?
I missed it.
And so if you look further down in the research, Kiki, I think there's another video where you can kind of see it in real time out in the field. They also analyzed pictures of the ripening of those fruits to measure the curvature of the stem. So that just happened. I don't know if you saw that. It was so fast. So they touched it and nothing happened.
I've seen these plants before too.
Yeah, and then it kind of touched a little harder and
wow. Yes.
Yeah, absolutely.
It launched itself at the ground while it did that.
Yes, but the seeds, importantly, went up. They can achieve speeds of up to 29 miles per hour and reach shooting distances of up to 12 meters. It's like 36 feet. They also noticed that the fruit stems straighten up during ripening. They create about a 53-degree angle. The theoretical perfect angle for this explosion is 50 degrees. That would maximize shooting distance. So the fact that they go to about 53 is pretty good. They always exit the seeds facing the same way. And there is a mucilaginous coat. Say that 5 times fast. When they get wet, which becomes adhesive when it dries, and so that they can get stuck to things, but also it improves the conditions for germination. So they think that, most likely this is about dispersing seeds in the most efficient way while not destroying the whole plant too early. So they want to make sure it doesn't happen until it's exactly the right moment. And so there's this huge evolutionary drive for plants to disperse their seeds as far as they can, of course, like we discussed. Now, why do researchers care? Well, aside from just basic ecology knowledge, which is always good, of course, I don't know if you can guess it, Kiki, robots. They want applications for bio-inspired launching systems as hydrogel-based actuators for medical tools and micro robots.
Interesting. Okay, so for medical actuators, that's fascinating.
Yeah,
mean,
they also think there's some potential applications for soft robotics like drug delivery systems
mhm.
where energy efficient launching is desired. So I don't know if I want that explosion happening in my body, but
Depending on what where and what's yeah, right?
at what magnitude, how tiny is it, right? Yeah, anyway. But yeah, exploding cucumbers, you're welcome.
Thank you for the, explosive seed dispersal story. Speaking of
Yes.
exploding no, not exploding at all. Let's talk about zoonoses. Don't
Hopefully not exploding. Yeah.
oh my gosh. There was a point in time when I was young and did not understand the science of, hemorrhagic diseases very well, and the sci-fi horror vision of, like, a person just exploding everywhere was
Hmm.
kind of I did have that in my head. No longer, no longer, everyone, because it's not really that's not how it happens. But how long has humanity been affected by zoonotic transmission of diseases, human pathogens that have, not just been human pathogens, but came about because of us hanging out with animals and that they joined us thanks to the animals. Well, these researchers publishing in Nature Open Access this last week have shown their analysis uh, of human DNA that goes back 37,000 years. And so the breadth, I guess, the distance over time that we now have in our ability to see what diseases have been impacting humanity for millennia. Now they have got these study samples that they've been able to take a look at. And although they go back 37,000 years, there's a lot more disease the closer we get to now. And specifically, in the data sets, the most interesting aspect of the work that they're reporting on is the fact that there's an uptick in the amount of disease that happens just about at 6,500 years ago. What happened around 6,500 years ago? Do you know? Do you remember, Blair?
Well, God created the earth. No, that's not right.
No. Oh my god, I just ugh.
No, I don't know. What happened 6,500 years ago?
So about 6,500 years ago, what we saw was the advent of farming and
Mm,
of keeping animals,
sure.
ranching and farming, and people settling down and becoming less nomadic and becoming more settled. And as that happened, there's a massive uptick in the time source of microorganisms and how they impacted humanity. They were even able to see like upticks through their different microbes that they were looking at. They were able to make out infections. They found that the oral microbiome was a great source of data for all of their work that they've done. Um, but this isn't necessarily the stuff that we're dealing with now, which is animal, uh, you know, going the live animal markets in very populous regions, which very likely is why we have a lot going on right now. But historically, we didn't have as many people. There wasn't as much density. We increased density and overlap of populations more and more
Hmm.
when we really got into the farming and trade that relied on that. So, anyway, yeah, 37,000 years of human disease history, and about 6,500 years ago, the really interesting bit happened, and, kind of tells us a story of,
Hmm.
you know, how we got to now. These are
Well, it wasn't the dogs then, because we domesticated them, or they domesticated us, depending how you want to look at it, way before that.
exactly. Well, those the
So,
dogs were
yeah.
domesticated. We were still very we were still nomadic in
Yeah.
many places. There was a lot of uh, there was still smaller communities hunting, very tribal as opposed to um, domestication, which is
I
why.
think that's really interesting that it took that because we're pretty young as a species in evolutionary time compared to other animal species around us.
Yeah,
So it would just kind of be my assumption that there would be a lot of zoonotics from day one.
I imagine
But I
that.
guess it's all relative, right? So it's just
Yeah,
so much more now. It's more than most other mammals, probably.
yeah, and so it's of course there are zoonotics is dis- are other, right? It's these microorganisms that are not from us or not just between us. And so, of course, as we're moving around, there were infections that occurred. I mean, there's research we've covered that suggests herpes virus has been with us for tens of thousands of years. Going way,
It's
way,
the
way, way back.
perfect disease. It doesn't kill you, and it's transmitted via sexual activity, and you can't always tell somebody has it just by looking at them. It's perfect. It's
It's
been made to do its job perfectly.
Yep. So there are things like herpes virus that have been around for a long, long time, right? But like, okay, at one point it was zoonotic. Now it's endemic. Like now it is just all it's a human disease, right?
Hmm.
And the plague, so bubonic plague and Yersinia pestis is another one that they tracked, and they saw that pop in and out. And there was actually a nice little period of time when there was no plague in the human populations, and then it whoop, popped right back up again, and then we had dark ages and the oopsies. Um, but it's an interesting look at a lot of very, very interesting, using the technology that they've been able to adapt to show us this, what they say, spatiotemporal distribution of pathogens in ancient Eurasia. So going back across Eurasian history, to look at bacterial, viral, parasite DNA, um, and while a lot of them, like they said in this study, are known human pathogens, the ones that were interesting are the zoonotic ones. So which are the ones that are coming in because of maybe our our close nature with other animals, and how did that lifestyle change impact us then, and how is it still impacting us now? Woohoo! Humans. It's cool. Zoonoses are awesome. They're
Hmm.
great. They're
No, they're
totally
not. They're not great.
And just like a sideline on another DNA study, researchers did publish this last week of a full genome that was, sequenced from a man in a pot in ancient Egypt. So it was the full remains of, an individual from ancient Egypt, and they were able to sequence the DNA 100%, human genome sequence to be able to say, "Where did he come from?" And basically, it was like, "Oh, yeah, he came from, like, the Egypt area."
Egypt?
Yeah, but
Thanks.
but there was about 20% of the genome that was attributable to ancestry in the Fertile Crescent. So,
Hmm.
the Eastern Fertile Crescent across the river.
That's not that far.
It's not that far, but it's the idea that, no, of course, they weren't, and this is again, in the, uh, that time period, it's probably about 5,000 or so years ago,
Kiki,
4,500,
they
five
parted
thousand
the
years
Red
ago.
Sea. You know about this.
I don't know, 6,500, whatever, 6,500 years ago?
They parted the sea. They crossed over. It's all good.
Yeah. Anyway, they crossed over. They made friends. And so dude found in a pot. He's like, he's a mixture.
How big of a pot are we talking?
An
It
urn,
was just,
a
it was
vessel.
human soup, basically. Was
gonna-
it dry or liquid? This is what I need to know.
Okay, let's see, the news, I hadn't actually, because I was hoping that Justin would be here tonight and would bring this story,
I
'cause
thought you were going to say, I was hoping you were going to ask me any follow-up questions.
no.
Sorry.
I'm gonna share a picture now of, this paper from Nature, and this is, the paper's whole genome ancestry of an Old Kingdom Egyptian. 4,500 years ago, give or take a few years, and they were able to look at the pottery vessel in which the individual was discovered. They found cervical vertebrae within the vessel as well. So that vessel, not the vessel, but the vertebrae showed evidence of extreme osteoarthritis,
Mm.
which is interesting in itself. But the bottom line to this study of the size of the vessel, it was a mummified, preserved individual, and it was in this ceramic pot was interred in a rock cut tomb. And because of the preservation, they think that also, the way it was interred potentially allowed it to be very well preserved DNA as well. Anyway,
But Kiki, I must know. Olive oil or paprika? What are we talking about here? Is it liquid or is it powder?
in the Blair,
These are the hard-hitting questions
it
that you
really.
come to This Week in Science for.
Okay, all right. Okay. I'm reading the paper right now. Okay, old Egypt, early dynastic, old kingdom periods, third and fourth dynasties, body in a large pottery vessel,
Mm-hmm.
probably from a higher social class.
Okay.
That they predicted that this individual had brown hair, eyes, skin pigmentation ranging from dark to black skin, genetically male, had osteoarthritis, probably was about 157 to 160 centimeters tall, lived between about 44 to 64 years old, heavily worn teeth,
Cool.
osteoarthritis. So
Yeah.
we've got solid bone
Okay.
in this urn. So
Okay.
I am going to make an assumption, and I would like anybody to help me with this. These remains were most likely, if they're interred, somewhat mummified, that the liquids have been removed from the
I
body
see.
so
A
that
dehydrated stew.
yes, but still with stuff.
I don't get it. I feel like I have to do some research for myself just to understand. I'm not my brain's not connecting the dots here of what was in this pot. Like how do you collapse down a whole human into a large pot? I'm not getting it.
Oh, this is okay, hold on. I have another image. I have a new image added to the stage. So here we have
Oh.
archaeological site data where we have
Oh,
hillsides
that is a
in turn,
large pot.
and it is a large pot. This is not a small coffee mug contain, not a
That's less
coffee
of a
can.
pot and that is more of a coffin, I have to say. Like
It's
that is yeah.
yeah, it's a pottery coffin.
Uh-huh. Uh-huh.
Yeah.
It's an ovular coffin
Yeah, it's
for the
a
fetal position.
it's like a very large stew pot,
Yeah.
but that's pottery and is filled with desiccated,
Yeah.
dried, preserved remains.
Yeah. Okay. That makes way more sense. Thank you.
You're
Thank you for
welcome.
digging.
I'm glad that I was able to find this.
I just have to call up the
Like-
researchers. Hey!
I was like,
Explain this to me immediately.
because it's not what you would think of as it's not a sarcophagus. It's not it's not that the individual like in modern human customs where we will you know have the remains burned and ashes put into a small urn that fits on top of your mantle.
Mhm.
This would be in the fireplace. Or
So
this
the
would not
original
fit on a
picture,
mantle. Yeah.
the original picture didn't really have any context for size, and so I was picturing something almost like a Le Creuset, like Dutch oven situation. Like that's what
I
it
kind
looked
of was
like, right?
I kind of was too. Yeah, and I was like, your question is very valid, and this is why I went silent at not having that information, and I need
Wow.
inquiring minds, we need to know.
Yeah. So I think this is also a really good example of researchers knowing exactly what they're talking about and not giving the right context.
It was in a pottery in a pottery urn.
Yeah.
Yeah, no,
Yeah.
this is a very
Wild.
and
Okay.
this is a full-size individual who they with their extended data, like they think that they have been able to actually do some reconstructions and
Hmm.
from what and the majority of the skeleton was there, and it really is because of the teeth and the DNA, then the dentin, and the preservation of those remains allowed us to be able to look at this individual and go, we know there was back and forth across that fertile crescent, east to west. Yes. This is This Week in Science. Thank you so much for joining us in this exploration of the unknown, the things that we would like to know. Sometimes we don't even know the questions or the stories to tell until we don't know the stories until the questions are asked in the right way. Sometimes the information and how it's told so interesting. It's such an essential part of the whole thing. So we're glad you're here for the entire process. Thank you for joining us. If you are interested in being a supporter of This Week in Science in an ongoing fashion, one of our non-corporate sponsors, because we are an independent podcast, science talk show broadcast on without corporate sponsors telling us what to do or how to do it, when to talk one way, when to talk another. It's you. We get to hang out with you. And thank you for doing that. Head to twist.org. You can support us on Patreon if you click that Patreon link. And I can't wait to read your name at the end of the show when you become a Patreon supporter. And if you're looking for fun items or new ways to show your love and support for TWIS, click on that Zazzle link as well. That Zazzle link has a bunch of products, and some of them they don't even look like they're like branded science-y kind of stuff because they're Blair's Animal Corner art, and they're very cool. And the pillow bolsters you should have on your couch, and your shower curtains, and the towels. I mean, come on, people. Everything goes to support This Week in Science. Thank you very much for all of your support in all that you do as a member of our audience. We thank you. We can't thank you enough. And now we will move back to more science-y goodness. I don't think there's any more exploding seed dispersal, but it is time for Blair's Animal Corner with Blair.
She loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant
What you got, Blair?
I can't believe I have to say this in the year 2025.
What?
But tortoises have feelings.
Of course they
Yeah.
do.
Of
Oh,
course they
okay.
do.
Tell me more.
Animal behavior and cognition experts at University of London, UK, looked into reptile sentience and reptile emotions. They put forward compelling evidence that red-footed tortoises, Chilonoidis carbonaria, sounds like pasta, experience long-term mood states in a similar way to mammals and birds. They studied 15 red-footed tortoises. They used a cognitive bias test, which is an approach that they used originally in humans, which reveals whether individuals respond optimistically or pessimistically in their judgment of ambiguity, depending on their mood. They've used this in mammals and birds mostly, but this evaluates how animals interpret ambiguous situations, acting as a window into how they feel. They found that tortoises housed in enriched environments, so environments where there's lots for them to do and they are happy and they're well taken care of, they were more likely to judge ambiguous situations optimistically, which suggests a positive mood. They also looked at their behavior in anxiety-related tests, which involved exposure to novel objects and unfamiliar environments. Tortoises that showed more optimistic decision-making in the cognitive bias test also displayed less anxious behavior in response to novelty, reinforcing the link between mood and behavior. This adds to a growing body of evidence which suggests that reptiles have more complex inner worlds than we thought. Who thought is my whole thing. Understanding whether animals can experience moods and emotions is crucial for informing welfare standards. In pretty much everywhere, reptiles aren't held to the same standards of care as pets than birds or mammals. And so the hope is that studies like this will change how reptiles in captivity are managed, how they're regulated, and what standards of care look like. They also say further that if reptiles, a group that diverged from mammals and birds hundreds of millions of years ago, can experience moods, that would suggest that the affective states are more widespread in the animal kingdom than previously believed. What?
Things that relate to survival, mood, emotion, etc. Like it's not all just stimulus response behavior, right? But you gotta do the work.
But think about it. We know that plants get stressed out when you repot them. And sometimes plants will look like wilty and gross for a couple days, and they're like, all right, I'm getting used to this. And then they perk up just fine.
Mm-hmm.
If and plants...
tortoises are long-lived.
Yes.
Decades. Over 100 years.
Like an animal with social structures, with interactions with one another, with complex lives, with long lives, like you're saying.
It's the long life. It's like elephants.
Any animal with agency has got to have a mood. You can't... Animals feel.
Yeah. And yes, and I agree. And this is where I've always agreed with you on, it's the well, duh side of things. But from the absolute, I know nothing, I'm an unbiased scientist perspective, you have to break it down into all of the questions, right? What is the question you can ask? And this is the balance that I think is part of the disconnect between like society and a lot of science is that absolute, just cutting, breaking everything down into its most discrete parts before you can accept a learned experience.
I think there's implied context that needs to be explained better, right? Like,
Yeah, yeah.
scientists aren't saying, we thought tortoises didn't have feelings. But because it's
News
presented
at
such
11.
as previously to now, there's
Mhm.
never been evidence that reptiles have feelings. Like, that sentence means different things to someone who is deeply immersed in science and eats, breathes, sleeps
Yeah.
scientific terminology versus the average person. Because to a
Mhm.
scientist, that means we haven't tested this yet. But if you say there's no evidence that, to a layperson, that means we don't think it's true.
And that person who has maybe gone, lived places where they've interacted with the
Right.
herps, the
Mm-hmm.
reptiles, the fish, the
Mm-hmm.
bird, all of the other, and they go, "What? Scientists don't know anything." Yeah,
It would
so
be
it's
great if there was just the context of, hey,
yeah.
we know reptiles probably have moods, just like any other animal, but it hasn't been tested. So let's see to what extent reptiles have moods, how it impacts their behavior, and if there's a long-term kind of trend for individuals in their moods. But that's
And
not how scientists talk.
I know, but now I'm thinking about, like, these gazillion decades old parrots and elephants and tortoises and sea turtle, all of the long-lived animals on the planet, and I'm like, okay, let's look at what are the commonalities in their cognition? What are the they're from, Aussies to have a different brain structure than the reptilian brain, which has a different brain structure too, like they're really interesting functional and structural differences, and I wanna know if an elephant is thought to have a long memory, what about a tortoise,
Hmm.
right? Yet but then
Yeah.
elephants, we know elephants that they hang out with other elephants, they got their big group. Where do the tortoises there I think of tortoises as kind of individuals and independent. Do they
It
have like
depends
their
on the
yeah,
species. Yeah.
okay.
Some of them have kind of little cultures and kind of colony style living. Absolutely.
I wanna know more about all that. That's
Oh
awesome,
yeah.
amazing. Okay.
Yeah.
So
Anyway.
so cool.
Well, speaking of feelings,
Feelings, yes.
I want to talk about hermit crab feelings. And specifically, when I say feelings, I mean feelings.
Like touching feelings,
Like
not
literally touching things, feelings. Yeah. Hermit crabs, they're constantly monitoring their environment for changes, for threats, for predators. When they detect danger, they retract into their shell. And after the initial shock of being startled, they'll use their sensory organs to determine if it's safe to come out and explore again. Because ultimately, that's what they have to do. They have to go find food, they have to go find mates, they have to go find a bigger shell sometimes. There's plenty for them to do out in the big, big world. In one local species of hermit crab, this is local to the UK coastlines. I love how they say that. They don't clarify. So this is near University of Plymouth. They have a local hermit crab that has tiny hair-like structures, sensilla, that's what they're called, on their claws. And that has to do with how they gather information about their surroundings, including the presence of potential predators. In this study, they actually found that crabs that have more sensory hairs on their claws recover faster from a startle response. So actually, the kind of more metaphorical feelings are also involved here, based on their sensilla and what they can physically feel and sense in the water. How they feel it might be safe or not safe to come out is as a result of that. So animals recover faster from a startle, and when they do, they're considered bolder. And in this study, those animals had more little hairs all over their claws. So researchers were even able to predict the time it took them to recover from the fright based on how many sensilla they had on their claws. Now, I bring this up also because I've brought a couple horrifying research methods to TWIS lately, and this was a rare situation where researchers actually took great care of the hermit crabs they were studying. They wanted to see how they used their claws and other sensory appendages like their antenna in exploration. And so what they did is they took some hermit crabs into the lab. They analyzed how they responded to being startled. And then they waited for the crab to shed their exoskeleton. They just waited.
Which they do naturally. They don't have to
Yeah.
force it. But so
Yes.
do these sensilla, which are
Mm-hmm.
kind of like whiskers or
Mm-hmm.
the sensilla on
Absolutely.
cats
Yeah.
or mice or so many organisms. Do they stay in the fleshy part of the animal as the
Mm.
exoskeleton comes off, or
Mm-hmm.
are they like really part of that hardened exoskeleton?
They appear to be part of the hardened exoskeleton because when they collected the molts, they looked at the shedded tissue and they looked at through a scanning electron microscope, and they were able to see the sensilla still on their claws. They were able to mark those sensilla on the surface, and then they were able to count them. And they didn't have to do that. They didn't have to remove limbs, which apparently is how that often is done in these studies because they want to collect everything at the same time. So they just remove all the limbs.
Yeah.
But in this case, they waited for them to shed and then they just counted what they saw. Then they assessed the number of sensilla related to boldness of the crab. They found that bolder hermit crabs have more sensilla on their claw surface. They also found that more sensilla suggested a crab had better access to information, which appeared to make them more consistently determine that the surrounding environment lacks risk. So basically, they came out sooner because the
Mhm.
extra little hairs told them, nah, it's good. You're good out there. There's nothing out there. It was just like a weird wave. Come on out. And so this brings a new hypothesis about how sensation and animal behavior are linked. And so they have termed this sensory investment syndrome. And so
Ha!
they hope that this will inspire other work which examines how sensory traits shape animal personality and decision making. So if sensory investment helps explain personality in hermit crabs, of course, that might be the case in other animals as well.
I think this is fascinating because you've talked about previously, personality types, quote unquote, in mice. We've talked about it in fish, and I don't and we also have, like, when we talk about human sensory abilities, there is a spectrum of being highly sensitive to things like touch, to being a super taster. Some people are, have extra chromat vision, right? So there's the sensory aspect of what it allows you to sense. What do you sense? What is normal? What is not normal is different. And this is
Yeah, so are the
I love
super
it.
tasters like thrill seekers also?
Right? I wanna know the same kind of thing about, like, we're asking about this, but, like, the number of, like, little whiskers that are on your cat or the whisker
Mm-hmm.
hairs, right? The sensory hairs that are on your cat or your mouse or your rat or whatever. How do those relate to this correlation of
Mm-hmm.
boldness or I don't know, your thrill-seeking dog? I don't know.
Yeah, absolutely. You're like, is my dog dumb or are they just a thrill seeker?
But yeah, and this is correlation also, and so there's
Yes.
probably
Yes.
other factors involved, but
Mm-hmm.
this is really interesting.
Yeah, like it's totally possible that they just got better resources when they were originally growing, and so
Yeah.
they got a better brain and more whiskers on their claws, and they're unrelated.
And maybe if because they collected the hermit crabs and kept them in a nice environment until they shed their exoskeletons, maybe more sensilla, like, weren't
Yeah,
broken, were
absolutely.
there's so
Yes,
many little but
there was in true Justin fashion, I will tell you, there is a story that I did not bring tonight. And it was about how rats with extra long second fingers, I think it was, were more sexually active or
wait, like the
sexually interested.
testosterone hypothesis?
Yes, yes, it is exactly that. Yeah. And I was like, I don't have it to get into this tonight. But yes, there's some excellent correlation.
Yes.
It's that whole book of correlation where it's a number of deaths in swimming pools versus number of Nick Cage movies released that year, right? Like it's just things correlate. That doesn't mean they cause each other, right? So it's
Exactly.
Anyway.
And we're going to say it over and over and over again. And it's fun to,
Yeah.
studies like this, I think, are very, very instructional. They're informative, right? But they are not the
Yeah.
end all be all because we don't like, and here is the scientist part of me that is, paring it down to the ultimate question, which is for each of those sensilla, what are they detecting and what is the pathway and what is the
Right.
integration in the brain that leads to that
Mm-hmm.
particular behavior? So there's
Yep.
a chain.
Yep. And if this is the sad part, if you pluck a bunch of them off, are they suddenly less bold?
Well, that's the experiment, right?
Yeah. Or if you like paint them with nail polish so they slick them all down or
Well,
like
yeah, so they can't use them. Yeah.
mhm.
I mean,
Mhm.
like
So,
in the same individual, like that would be a good
mhm.
study. Hey, grad
Mhm.
students, there's some good studies to do. Be
No
nice
money to do it, but maybe somewhere else, maybe in the UK.
because yeah, I'm sure there's private equity to study hermit crabs.
Yeah.
Anyway,
Kiki, you had a weird animal story.
I do. I do have a weird animal story, and it's just one of those studies that I'm like, am I really gonna tell this story? Um, all right,
You
so
tried to make me do it, but I
I
didn't have
did.
time to read it.
I did try to make you do it. Okay, so this is still Animal Corner, which is why I was
Uh-huh.
wanting you to chimpanzees at the Chimfunshi Wildlife Orphanage in Zambia have started fashionable trends that the youngsters are picking up and repeating and seem to be doing this. Once upon a time, back in 2014, researchers reported what they called the grass in ear behavior. And it's a very simple behavior in which the chimpanzees don't take a lot. It's not like grabbing a tuft of grass. It's a blade of grass and just putting it in
Hmm.
their ear and letting it hang there for a
Mm-hmm.
while.
Like a cool earring.
It is like a cool earring, and
Hmm.
the chimpanzee in this particular image, by Jake Booker from the, Chimfunshi Wildlife Orphanage Trust, the chimp looks very, very cool. Um, so there's a new study that was just published this last week, 4th July, in Behavior, and it's about chimpanzees learning their just this kind of doesn't what function does it have kind of behavior, and it's called chimpanzees socially learn non-instrumental behavior from conspecifics, which means that they're learning from their friends. They saw they saw others do things and then started doing them themselves, and now we have, according to their abstract, over a decade later, they've observed an unrelated group of chimpanzees at the same African sanctuary where 5 out of 8 individuals began wearing grass in their ears, and 6 out of 8 from their rectums within a short period of time.
Mm-hmm. Mm-hmm. Mm-hmm.
And neither of these behaviors have been observed in any of 7 other sanctuary groups that they have. It's like this one particular group within this sanctuary, except for 2 males who were in, I guess, the original grass in ear group. And so they've concluded that these chimpanzees have learned from each other, and it's a social web that is has led to, yeah, the social web that has led to the development of this behavior.
So I like in this article, I'm reading ahead, I'm seeing that there's a theory that the caretakers had something to do with it because the caretakers sometimes stick things in their ear to get things out.
Yes.
Or what I was thinking was it might be an earring if some of them have earrings, right?
And that would be
But
like
yeah,
that would be where it first started
yeah.
over a decade
But
ago, but
but
now they have
then
taken it not just from the ear, but to other orifices.
yes, which I don't quite get that one.
I
They're
and
just experimenting, I guess.
I think so, but they're seeing others experiment and they go,
Yeah.
"Ok okay."
I guess it's fun. It's like a tail or something. I don't know.
And maybe that's part of it. I mean, we have absolutely no idea what is going on in the minds of these chimpanzees. We see that they are that the community structure definitely has influence on the behavior and the innovation and the time in which it takes to spread um, from individual to individual. And so we've got, over time, from the innovator to the individuals that that innovator is connected to, suddenly you have more experimentation and play or use. And it's not necessarily a behavior that you would suggest has a, a purpose other than maybe
Hmm.
social connectivity. So,
Yeah.
but anyway,
Hey,
chimpanzees.
listen, they don't have the shame that we've manufactured for ourselves. They're just trying stuff. All the power to them.
Chimpanzees, they're like people, just no shame.
They're just not repressed.
But, yeah, anyway, I thought it was a very interesting study. I
Yeah.
I thought that if had you had more time to dig into it, there might be more that you would say, but I don't know.
Have fun, guys. That's what I have to say. You're not hurting anybody.
Glad you're having a great time there, yes. Okay, as we end the show this evening, I've got 2 stories for my last segment. Thankfully, as an adult, research has confirmed I've got new brain cells, and you might too. You probably do too.
I need some. I lost so
Right.
many.
I know. So, for years, the question has been, does the adult brain continue to develop and birth and, propagate new neurons? Because it's something we do when we're young, and once upon a time, it was like, "Hey, you have all the neurons that you're ever gonna have when you're a baby," and it goes downhill from there and never gets any but then you think about it, and it's like, "Well, that doesn't make any sense." And we've seen evidence of new neurons being born in birds and mice and all sorts of other species, and so, the problem is you can't just cut up people's brains while they're
Hmm.
alive to find out whether or not,
Why not?
"Oh, darn." Um, yeah, well, that's a thing that hopefully scientists have developed and are continuing to develop, which is ethics. When it comes to research,
Yeah, we'll see.
especially involving people and animals. I mean, it's very important. This work is out of the Karolinska Institute in Sweden, and the researchers have for this study in which they really do address that question of is there continual adult neurogenesis in the particular area of the brain that I love, the hippocampus. The hippocampus is like if you were to take your fingers and kind of stick them toward the middle of your brain. It's in there. It looks like a little seahorse, which is why it's called the hippocampus. It is an area responsible for much of the learning and memory that that our brains are capable of. It is the area that's kind of like the librarian. It takes information in and puts it out and connects it to other stuff. The hippocampus has been shown in so many organisms with incredible specialized memories to have neurogenesis, which is, the birth of new neurons. And so we've been like, wow, okay. Well, we've looked at the brains of people who have died. We've looked at this. We can't, but it was always correlation. And so now the big question was, can we actually identify proliferating neurons in the hippocampus? And through looking at the traits of proliferating neurons in other animal species that we have identified through other methods historically, and using brain scanning and mapping of the brains of deceased who had donated their brains for science, as well as machine learning algorithms, they were able to analyze the human hippocampus from birth through about age 78. So, birth through adulthood. They used single nucleus RNA sequencing. They were able to identify all the neural progenitor cell stages in early childhood. And in adults, they were able to use antibodies for particular markers and these machine learning algorithms and identify proliferating neural progenitor cells, which means they were dividing. They were creating new neurons. The neurogenesis was ongoing. So, yay!
That's great. I need it.
Yeah,
Hurry up.
yeah. They show that the end in the hippocampus, the particular area that these progenitors were localized in is the dentate gyrus, which is, yeah, go look up that one, everybody. Go learn about the dentate gyrus. I would like a 10-page essay on it on my desk by next week. Hopefully not written by Claude or anybody else. And then finally, we've talked before about the wonderful feeling of being in a moment with other people. And we've talked about how when you're watching a TV show, there's like sometimes an engagement, like your brain synchronizes with activity in the show. If you're, there has been work that suggests that even Blair, you and I, like I am synced with you, even though I have crooked glasses, like I am synced with how you are and how we're interacting through this interface, even though
Mhm.
we are in distant places. And it's probably a weaker brain synchronization than it would be were we in person. But there's a synchronization of our brains going on. And the people in the chat room, people in the audience who are watching live right now, they potentially are synchronized with each other
Hmm.
and with us because of that togetherness of a live social event. And these researchers have published their work, which has been ongoing. That it's a European initiative called NeuroLive, and this is an ongoing effort. And what I love about the work they're doing is they're trying to do like EEG brain recordings of live performances, recorded performances, and all sorts of different ways of engaging with performances. Most specifically, they're using dance as the artistic performance that they've been looking at. And this latest paper that they are publishing on, they have found that the delta band of brain activity, which is a frequency of brain activation that is often related to social activation and also to like also your mind wandering. The delta band EEGs were synchronized with live and recorded dance. And so the very I think the interesting aspect that has come out of this is not just that it's, hey, we recorded everybody in a theater. while they were watching a dance performance, and everybody who was there live had brain activation that was synchronized. They also showed that when they had people together watching a movie of that dance performance, they were all synchronized. And the weakening of the synchronization happened when you had individuals watching something alone in a laboratory environment. But it was still the performance and the timeline of events and things that happen in that performance. They had the choreographer highlight which moments they thought were the ones that the audience would react to the most, and those were the ones that all the brains lined up to in the
Interesting.
synchronization. Yeah.
So next time I want somebody to go to a concert with me or a movie with me, I'm not going to ask them that. I'm going to ask, do you want to go synchronize our brains?
Uh-huh. I mean, it's like, do you wanna go? Like, it's like a dance performance, but it's really gonna be like a synchronized social experience for us.
I mean, it makes me reflect on the fact that we like to do these things as a social activity when you can't talk. And that's always kind of baffled me, is that I could talk to Brian forever, but we love to go to movies together. We love to watch movies together. We like to go to concerts. We like to go to live shows. And you don't get to talk during that time at all. But for some reason, you can still look back on it and reflect on it as an experience that you enjoyed together. You weren't just sitting next to somebody. Your brains were doing something similar, and that's like a bonding experience.
Yeah. It is a bonding experience, and the, you know, the interesting, important, fun part of this is that question that you're bringing up there, which is, can we or will we ever, and why why would we want to get rid of that live experience
Mm,
that you have together? There is something very powerful about the sync, and it's not, "We were there together," and of course, we've all known that. "Oh,
hmm
I wasn't at Woodstock," either of them, but people talk about those things, right? People talk about these certain experiences that you had, these concerts, these performances, the speeches, things that they experienced with other people, and we, in our lived experience, can say how it feels, but the fact that it's common that
hmm
the beats in the performance are something that every brain, more or less, I mean, of course, there's gonna be variation, but that brains are picking up on is is just fascinating. So, this research program has been ongoing, and this is the last year of its program, and, hopefully, the work that has come out of it and the papers that hopefully will still come out of it will tell us much more, especially given our experiences from the pandemic, from social distancing, from becoming ever more online, and what that means to how we are going to coexist as humans.
hmm
Yeah. Yeah. Somebody says there are lots of plots in this. There's all sorts of them. We've got, yeah. It, but the sitting alone watching a recording in a laboratory environment was definitely where it stopped working. Where where the synchronization didn't happen quite so much. But there's a lot of data in here. This is an open access paper, and so I think that if you're interested in this kind of stuff, that it's worth digging into. Um, yeah, and I think another thing just to say out loud as I'm looking at this infographic from the paper is that um, the synchronous activity in those delta waves is during the moments of higher engagement, and so during the moments where it's like people go blah, 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 and mind wander off into their own spaces, That's not the synchrony, but it's the moments in a performance that bring people together, right? The performer, to be or not to be, that is the question, saying words that pull you all back together, where there's something dramatic, whatever. And that's going to, create arousal and attention and psychological modes that lead us to where we are. I hope so. Kevin Reardon in the chat is saying the Greeks used to use theater as therapy. TWIS is not theater, but it's a little bit of therapy to me sometimes. So I hope you all feel that way as well.
Disclaimer, disclaimer, disclaimer. We are not licensed therapists or psychologists.
Oh, no, I meant for myself.
Oh, okay. Got it.
Oh, dear. Have we done it, Blair? Have we made it?
We've done it.
Oh, yes. All right. Everyone out there, chat room, those who are listening, those who are watching elsewhere, wherever, whenever you are, thank you so much for joining us tonight for another episode of This Week in Science. We do appreciate you being here. And thanks, as always, shout outs to all the wonderful conversations happening in the chat room. Gord, Arenlor, Fada, others who help keep the chat rooms nice places to have those conversations so that we can all chat as compassionate, respectful adults, human beings. Fada, thank you so much for doing the social media and show notes. Really, really wonderful that you're able to do that. Rachel, thank you for editing the show. Identity4, thank you for recording the show. And as always, I really must say a big thank you to our Patreon sponsors. I'm not going to go backwards this week. Last week was a whole thing. Thank you to Robert Norlin, Robert W. Farley, Lauren Gifford, Dana Lewis, Eden Mundell, Alan Viola, Aaron Anathema, Arthur Kepler, Craig Potts, Mary Girth, Teresa Smith, Richard Badge, Bob Coles, Kent Northcote, George Corris, Pierre Velazarb, John Ratnaswamy, Chris Wozniak, Vagard Chefstad, Donathan Stiles, a.k.a. Don Stylo, Ali Coffin, Shubu, Don Mundus, P.I.G., Stephen Alberon, Daryl Myshak, Andrew Swanson, Fredis 104, Paul Ronovich, Kevin Reardon, Noodles, Jack, Brian Carrington, David E. Youngblood, Sean Clarence Lamb, John McKee, Greg Riley, Mark Hessenflow, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Brappen, Richard Brendan Minish, Johnny Gridley, Remy Day, G. Burton Lattimore, Flying Out, Christopher Dreyer, Artyom, Greg Briggs, John Atwood, Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul Phillips, Shane, Kurt Larson, Craig Landon, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, Lon Makes, E.O., Adam Meshkan, Kevin Parachan, Bob Calder, Marjorie, Paul D., Disney Pecoraro, and Tony Steele. Thank you for all of your support on Patreon. And if there is anyone out there who is not yet supporting us on Patreon and thinks that, hey, that might be a nice thing to do, you can head over to twist.org and click on the Patreon link to become a monthly supporter or a yearly supporter. Yeah. Blair, what's happening? Let's see what's happening on next week's show.
We will be back on Wednesday at 8 p.m. Pacific Time, broadcasting live from our Twitch, YouTube, and Facebook channels.
And if you would like to listen to us as a wait, no, this is you. I need you to do.
I can do the whole thing. Want to
You.
listen to us as a podcast? Just search for This Week in Science wherever podcasts are found. If you enjoyed the show, get your friends to subscribe as well. For more information on anything you've heard here today, show notes and links to our stories will be available on our website. That's at www.twis.org. When you go there, you'll get a nice pop-up. You can sign up for our newsletter. We'll send one day. Not sure when. Keep you guessing. We love your feedback. If there's a topic you would like us to cover or address or suggestion for an interview, please let us know on one of our social media accounts, or better yet, send us an email. Just be sure to put TWIS, TWIS, in the subject line, or your email will be shot out of a cucumber at 29 miles an hour, up to 40 feet. And we won't be able to find it. It's going to be all sticky. It's going to stick to something. It's going to be above our head. We'll never know. Anyway, we look forward to discussing science with you again next week. And if you've learned anything from the show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.